Okay, in animal cells, we just said that cytokinesis occurs by a process known as cleavage. And what will happen is there will be a cleavage furrow that forms. Now, at the top of this slide, you can see that there is an electron micrograph showing the cleavage furrow. Some people have said in the past that looks like an older person's bottom, but it isn't. Then what will happen is microfilaments, and again, here is another component of the cytoskeleton, will actually form in the middle where the cleavage furrow is and will eventually pinch apart the daughter cells. In plants, there is no cleavage furrow. Instead, they have what is called a cell plate, and that forms during cytokinesis. And what you can see here are vesicles actually move to the middle of the cell and start to form the cell plate. And later on, that will become a new cell wall. I'll show videos through Edmodo. Evolution of mitosis. The last thing I want to say is that um, there are different mitosis variations that have occurred. And one of the major ones that I want you to know is called binary fission, and that is the mitosis in bacteria. Bacteria only have one chromosome, and it is a circular chromosome. Remember, they have no membrane bound organelles, so they don't have a nucleus in which to hold many chromosomes. Um, right here you have an origin of replication, and then what will happen is there will be replication that occurs and it will produce two identical rings that are chromosomes. Then what will occur, and this is so much simpler, is the cell will actually elongate, and in the middle will be a ring of proteins that will then eventually pinch apart the two cells. Now, it is believed that there were some intermediate types of mitosis, and you can look at this. The very bottom is what we talked about through this whole video. At the very top is what I just talked about. And then in between, you can see that there are some variations that have occurred. And that is the end. If you are done with your video, then you can work on your chapter 11 maps. You can, if I'm done with the tests, which I may not be, you can work on your um, correctives or other things that you need to do, but there's plenty that you could be doing. Thank you.